Okay, so here we are. We're going to talk about addictive drugs and get into some of the mechanisms. But first, I want to talk just a little bit more about this conceptual model in addictive drugs. And first, we should probably define addiction, right? And so this is the typical definition for addiction has to do with having a dependence on a particular substance. But really, what we should do is make it, because a little bit more, but substance suggests something that's tangible, something that you can hold in your hand or feel, something that's material. But I'm going to say stimulus instead, a dependence on a stimulus, which can be a substance such as a pharmacological drug. And this is the important part, because we have dependencies on lots of stimulus like water, food, oxygen. But the salient part here for addiction is that there is an inability to stop, even when there are negative consequences or adverse effects. So essentially, addiction is an inability to stop something, even when that something is hurting you. Even when you know this is hurting me and I don't want to do it, but you can't stop. That is addiction. And that's an important point to make. It's more than just, I want to take this because I want to take it. It's, I don't want to do this, but yet I'm still doing it and I can't stop. So you can imagine, if you've never suffered from addiction before or known anyone to suffer from addiction, this very much is a disease, like any other disease, any other in physiological disease, for that matter, as well. I'm not just talking about diseases of the mind, so to speak, or psychiatric diseases, but just medical diseases. Physiological diseases as well, because at the end of the day, this is all just molecules interacting with receptors, and the output is, in this case, a behavior, because it's taking place in the brain. But it could very well, something very similar could be happening somewhere else, and we could be talking about high blood pressure, for example, or something like that. But no, we're talking about a behavior, about addiction. And so addictive drugs, they bypass this black box that I described before, brain processing information, and it leads to an automatic increase of dopamine, which of course goes along this pathway here, increase of dopamine to both of these brain areas, the nucleus accumbens and the medial prefrontal cortex, remodeling for craving, and increased memory for the associations, the cues, and the behaviors. And what drug use can do, addiction to these substances, is over here in the memory side, these associations. What it does is this remodeling places a very intense maximal ability to remember and associate various non-drug cues with the stimulus that you're being remodeled to crave more and more. And this is the important difference here, right? Where the drugs, whereas other natural stimuli that you come across, you eventually cut out this first part here, the brain processing information, as it's no longer unexpected and you know that it's pro-fitness. The brain processing information eventually turns the VTA off and says, okay, we've interacted with this person before, or I already know what ice cream tastes like, and we know how to get it. It's at the grocery store, and I can get it anytime. So it no longer tells the VTA to remodel these areas. You don't need to increase the craving or increase the wanting, or increase your ability to remember where it is, because you figure that all out now. Drugs don't do that. They go straight for the VTA, so there's no turning it off. So every time you take the drugs, the VTA increases dopamine every single time you take the drugs. Now, the euphoric effects of the drugs themselves are not due to the increase of dopamine, but other things such as the release of endorphins, opioids in the brain. Even if they're not opioids themselves, the drugs not themselves. But the increase of dopamine is something that's separate from that hedonic signal, separate from the subjective euphoric experience. The dopamine is what is causing the cravings, what's causing the increased importance on the associations, such as paraphernalia, places, people. And to the point where this signal increases so much, but especially this one here, where you become so remodeled, the neuromodulation hardwires you so hard for these associations that these associated cues, the paraphernalia, the places, the people, can lead to an almost involuntary control of using the drug. So this can displace voluntary control. Right, this, right, and this craving and wanting can lead to displacement of the voluntary control. And if you listen to people who suffer from addiction or former addicts talk about this, even those that have been been engaged in sobriety for years even, they still talk about that the cravings are still there and that they have to be careful. In certain programs, one of the first things that you do is you separate yourself from the stimuli that you've learned, that have been strengthened in associating with the cravings and with the drug itself. You don't go to the same places. You don't hang out with the same people that are associated with that drug, because they're such strong triggers. And people will describe it as not having voluntary control. It's like having, I've heard it described before, as having an itch. And so you can imagine you have an itch that you really, really want to scratch. If you concentrate hard enough, right, you can control yourself not to scratch the itch, but it's hard and it takes a lot of concentration. But as soon as you don't focus on it, as soon as you're not concentrating on it, you'll scratch it involuntarily. And that's what happens. That's what's described often in like relapsing into drug use, is that it's just almost unconscious, that with being around certain cues and having your guard down and not being vigilant, it's as if you weren't even conscious of taking the drug again and falling off of the sobriety wagon, so to speak. Okay, so let's talk about some specific mechanisms now. Okay, so first we're going to need to talk about some of the neuromechanisms of our axon terminal and our VTA and dopamine release. All right, so this is the axon terminal. Of our ventral tegmental area neuron, so just one neuron. And so this is synapsing somewhere else, not in the VTA, right? But this is the axon terminal, so this means this could be in the nucleus accumbens, or this could be in the medial prefrontal cortex, one of those, right? And so first, let's just very briefly, I'll just write this out. We don't need to know the details. But dopamine is synthesized within the neuron itself. So tyrosine, if you can go back and look at the video and reference those, the synthesis of dopamine, tyrosine, gets synthesized into L-DOPA, which gets synthesized into, which is the precursor for dopamine. And dopamine will be packaged into vesicles. I'm going to draw the vesicle extra large here, just so we can better understand all of the little bits that are involved here. The first thing I'm drawing here is a transporter. This is what's known as V-ATPase. And that V stands for vesicular, right? Because this is in vesicle. And so this, I'll keep things red here. This is what's known as a primary active transporter. And what makes it primary? Well, what makes it primary is the fact that it uses ATP directly for energy. Hence the name. So it's a vesicular and ATPase. So it's using ATP to produce ADP and a phosphate in order to move hydrogen into our vesicle. We talked about this before in the context of not necessarily this mechanism, but in the context of the AB toxins, in understanding that the vesicles are, because of this process, creates an acidic environment. So it's more acidic on the inside, right? And that's what causes the dissociation between the A and the B toxins. So we have hydrogens in here. And so why do we have hydrogens? Well, it's involved in our need to get dopamine into the vesicle. All right, so there's another transporter I want to highlight here. I'll make it more obvious here. We have the blue transporter and our V-ATPase is red. Okay, so this one is, I guess I'll draw it over here. This is what is known as, for short, VMAT2. And what is VMAT2? That is vesicular monoamine transporter 2. Right, so vesicular, it's in the vesicle. Monoamine, dopamine is a monoamine, just like the other catecholamines, such as which is norepinephrine and epinephrine. Other monoamines include serotonin as well. So this is a monoamine transporter, and specifically the VMAT2. And this is what's known as a, well, here, let me describe how it gets in first before I talk about that. It's not a primary active transporter. It's slightly different, and let's understand why. Well, dopamine, the VMAT2 moves dopamine into the vesicle. But it doesn't do it passively. It needs to be actively transported. And so the way it does that is by transferring 2 hydrogens out of the cell. So it does 2. I guess I'll draw 2 of them here to make that clear. So for every 2 hydrogens, 1 dopamine molecule moves into the vesicle. And this can be done because the concentration of hydrogen is so high, right? Things are so acidic in here because of our VATPase, constantly using energy to pump hydrogen in against its concentration gradient. And this builds a concentration gradient where hydrogen wants to get out, so it sort of spins this wheel, so to speak, of the VMAT2, allowing dopamine to come in. Now, This is active transport because it's requiring energy, but it's requiring energy indirectly. The energy is coming from the VATPase, the ADP that's being used here, and it's storing that energy as a different type of potential energy in the form of a concentration gradient of hydrogen, which can be released out of the cell to move dopamine in. And when it's not using ATP directly, which is, I don't know if I said it before, but it's this process right here is what makes this primary, is because it's using ATP. But when it's not using it directly, we call that a secondary active transporter. Secondary active transporter. That is what VMAT2 is, a type of secondary active transporter. So great. This is how dopamine gets into the vesicle. And in action potential, as we know, we're very familiar with this now, and an action potential comes down, it stimulates the release by fusing the vesicle with the presynaptic membrane, and dopamine is released into the extracellular space. And so dopamine is floating around here. But as we know, there's an inevitable fate for neurotransmitters that are released, right? They can't just hang out there forever. We need something to get rid of them or bring them back in, and that's what happens in our cell here, is that we have a special membrane-bound protein that allows us to suck them back inside. And this is what we call simply the DAT. This is our dopamine transporter. Dopamine transporter. Great. So what this does is it will transport dopamine back inside the vesicle, right? And this can be, back inside the neuron, and this can be repackaged via the VMAT2 transporter, the VMAT2 same transporter, again, is redundant, via the VMAT2. And so the way it does this is that it allows dopamine to come in with 3 other molecules. 2 sodium ions and 1 chloride ion. And so we know that the concentrations on the outside of sodium and chloride are relatively higher than they are on the inside. And then also, in addition to the concentration gradients for sodium, the electrostatic gradient is higher on the outside. So it wants to move in doubly because of the concentration and electrostatic gradient. Okay, and so this isn't actually, I think I might have misspoke. If I called this little green dot, if I called DAT a transporter, it's actually, despite the name, it is called a dopamine transporter. But technically, it's what we would call a symporter. And any idea why? An active symporter. And that's because it's actively moving the ions and the dopamine in the same direction, as opposed to VMAT2, which is moving hydrogens in the opposite direction of dopamine. This is moving them both the same way, from the extracellular space to the inside, the intracellular space of the axon terminal of our VT8 neuron. Right? And so this dopamine transport, transporter is an appropriate name because you're taking dopamine from one side to the other, so it's transporting dopamine. But we call this type of transporter, it's an active symporter. because of the ions and dopamine are moving the same way. Okay, so now we've got this under our belts here, and we understand this sort of process. Now we're going to talk about the different addictive drugs that affect, and it's good to know these things because they're going to affect these different mechanisms in different ways. Okay, so now that we understand the cellular mechanisms of our axon terminal in our ventral tegmental area neuron and the release and reuptake of dopamine, now we're going to talk about how different addictive drugs alter these mechanisms here. So first we're going to talk about what we call psychostimulants. And the first one we're going to talk about is cocaine. So cocaine here I'm going to represent as just a pink circle. Okay. And so what cocaine does, and then what we're discussing here are the addictive properties of cocaine. Right? This is responsible for why cocaine is addictive. This isn't an explanation for the other subjective experiences that one has with cocaine. So just the addictive properties. And so cocaine, once taken, migrates to these areas, and it's attracted to our DAT, our dopamine transporter. Right? If you remember, this is what's responsible for taking dopamine from the extracellular space into the cytoplasm of the axon terminal, right? And it is I erased it, but it's a symporter. So it's doing this with 2 sodium ions and 1 chloride ion to take dopamine inside. But what it does is, by binding to DAT, it blocks it. So dopamine cannot be taken up into the axon terminal, into the presynaptic cell anymore. And so what we have here is dopamine remains in the intracellular, the extracellular space for a longer period of time. And what happens is it builds up because throughout our day, dopamine is constantly being released at a what we call a tonic concentration. And so I'll write that here, tonic concentration. And this is happening all the time. And so even without the release of dopamine, cocaine can drive an increase of dopamine concentration in the extracellular space, so it has more time to interact with the receptors in the target cells. So this is how cocaine influences dopaminergic neurons by blocking the dopamine transporter, preventing dopamine from being taken back up into the presynaptic cell. So that's fairly simple and straightforward. But let's talk about another class of psychostimulants that are a little bit more insidious. All right, and I'll start here on a new sheet. Let me erase this here. And again, we're talking about this again. These are psychostimulants. And what I want to talk about now are amphetamines. And I'll name a few here. Amphetamines. Some of the more well-known ones include, just simply amphetamine. We also have methamphetamine, which is just amphetamine with an extra methyl group. And another one that's relatively popular and well-known too, MDMA. And what is this? This is, better well-known popularly as ecstasy. And what is MDMA? This is, I don't have enough room here, do I? So I'll write it down here just for those of you who are curious. And so this stands for methylene dioxy methamphetamine. So let me spell that up for you. Methylene deoxy methamphetamine. Okay. And so these are three major amphetamines. Methamphetamine is more known as meth or ice. I don't know if that's a popular term anymore. I think meth is probably more well known. And so what do methamphetamines do in this scenario here? Well, something quite a bit different than our cocaine, and we'll make our methamphetamines, or amphetamines, orange. All right, so these make our amphetamines, these orange dots here. Okay, so what they do is, first, and so before I describe cocaine, and that's very well known, that mechanism, but this is a little bit more unknown, or at least to the best of my knowledge, in terms of how confident we are on these mechanisms. What we do know is you take amphetamines and you pour them onto the axon terminal of a dopaminergic neuron, and what you get is you get lots of dopamine. Lots of dopamine is sent out into the extracellular space. But what's interesting here is it does it without action potentials, and it does it without vesicular exocytosis, right? So this causes the release of dopamine. Let me write down here because this is pretty incredible. So amphetamines, let me change my size here. They cause the release of dopamine without vesicular exocytosis. So how the heck does dopamine get outside of the cell if you're not fusing vesicles with the presynaptic membrane? So let's take a look here. And so what amphetamines, what they do is they come over here to our DAT, our dopamine transporter. And what they do is they cause what's known as, they turn this into a reverse transporter. Right? So somehow, right, and like this isn't exactly well known, is what they do is that they can interact with DAT in such a way in which they can transport themselves into the axon terminal in exchange for a dopamine. So dopamine actually comes out, right? Reverse transporter. So it's reversed. The amphetamines get sucked up into the axon terminal, and dopamine is spit out, not through the fusion of a vesicle, but through the dopamine transporter working in reverse, working backwards. Right? And that's not all. It doesn't stop there. And so the amphetamines do the same thing to our VMAT2. So I'm just going to erase here the hydrogen. And so it transports itself via VMAT2 in exchange for dopamine coming out of the cell, out of the vesicle. So amphetamines come along and they spit dopamine out through the dopamine transporter, through a reverse transporter process, and they do the same thing, a reverse transporter process, by gaining entry into the vesicle and spitting dopamine out. And so this results in, taking amphetamines results in an increase of dopamine secretion, but without action potentials and without vascular exocytosis, without vesicles fusing to the presynaptic neuron. Okay, so let's consider a different type of addictive drug now.